Okay, so this video is going to discuss ion channels, action potentials, and contraction. So basically, how ion channels work, what action potentials are, and how it affects muscle contraction. Now I'd like you to pause the video here and play the video for resting potential that is also on the YouTube channel. Now the thing about that video is it does make reference to neurons, but what applies to neurons also applies to muscle tissue, so it'll still be good for what we're doing. Okay, so assuming that you just watched that video, I want to go into a little bit more detail about ion channels. As we learned for the for the second test materials, ion channels are basically transmembrane proteins. They're basically like a tunnel through the membrane. I want you to remember that ions can travel only through channels because the hydrophobic portion of the membrane keeps them from traveling through on their own. And so once channels have been opened, ions will move across the plasma membrane down their electrochemical gradient. And I want you to notice that I've written the term electrochemical in a couple of different colors. Blue basically for the electrical part, red for the chemical part. So let's take a look at how they move down their electrochemical gradient. First of all, positively charged cations are going to move towards a negative charge. Now I want to make something that's very clear. They're going to move towards a charge that is more negative than their immediate surroundings. So if a cation was around a charge of plus 100 and there was a charge of plus 30, that charge, they would move towards the plus 30 charge because the plus 30 would be more negative than plus 30 is smaller than plus 100. So a positively charged cation will move towards a negative charge. Negatively charged anions will move towards a positive charge. Now, when this movement happens, when ions do move down their electrical gradient, they'll create an electrical current, and this will alter membrane potential. So basically, if the inside of the membrane is negative 70 compared to the outside, and a lot of cations move in, you're going to have a lot of positive charges moving into the cell. That's going to raise the charge within the cell. Okay, now on a chemical basis, these are still chemicals that are moving from an area of high to low concentration. So this is just a diagram of voltage-gated ion channels. So as you can see on the left, there is a sodium channel. The sodium ions cannot get through because if you look towards the bottom of the diagram, you'll see that there is a blockage of that channel. This is called a gate. And so, excuse me, look at the charges. There's pluses outside the membrane, there's minuses inside the membrane. Okay, now if the membrane voltage changes, and we'll go into how membrane voltages change when we get into nervous system physiology, but if these membrane voltages do change, then you're going to see a difference in the change on the interior of the membrane. That's going to change the structure of the protein such that the channels actually open, and at that point sodium will come in through this channel and it'll actually come down its concentration gradient. Okay, now when I talk about a difference in charge in interior versus exterior, when we talk about a resting membrane potential, basically let's talk about a little bit of terminology here. So a potential means that one charge exists in one location and a different charge is at a different location. Okay, and so these charges are basically concentrations of usually either electrons or of ions. Now in biological systems, we don't ever see ions on their own. They always get taken up by something. So in physiology, these millivolt charges are related to ions. So they could be sodium or calcium or potassium, so forth and so on. Okay, so let's take a look at a little diagram here. So if this is a membrane, and we're just doing it real simple right now, if charge A is on the outside of the membrane and charge B is on the inside of the membrane, well, the difference between those two charges is the membrane potential. Okay, now the interior, in this case charge B, it's always used as a reference. Okay, so if charge B is 70 millivolts less than charge A, then you're just going to describe the membrane potential as negative 70 millivolts. If charge B was 90 millivolts less than charge A, then you would describe the membrane potential as negative 90 millivolts. So when you talk about membrane potentials, you are always talking about the inside as compared to the outside. Now, a question that I usually get when we go over membrane potentials is this. If we're talking about sodium, which is a positively charged ion, and potassium, which is a positively charged ion, how can there be a negative charge anywhere if we're talking about positively charged ions? Okay, so there's a little analogy that I have for this. I want to imagine that you have $100 and I have $30. We're both on the positive side in terms of money. So I've got $30, you've got $100, but compared to you, I have negative $70. And that's basically the way that this membrane potential is compared. So generally, in neurons, the inside is negative 70 millivolts compared to the outside because there is a smaller positive charge. Okay, now in terms of the cells that we have in our body, with neurons, the most typical charge is negative 70 millivolts. Now, there are always exceptions. There are some neurons that have different charges, different potentials, but for the most part, it's negative 70. And so that's the reason why that's the usual charge that's used in things like videos and when we learn about membrane potentials. Now, skeletal and cardiac muscle have membrane potentials that are closer to negative 90 millivolts. So they're a little bit more on the negative side than neurons are. Okay, so what I'll need you to do here is pause the video, play the videos for action potential and the propagation of action potentials. Now I want you to keep in mind, once again, this makes reference to neurons, the entire thing does, but muscle tissue has the same potassium channels, it's got the same sodium channels, it's got the same sodium-potassium pump, so what you see on this will actually also apply to muscle tissue. Okay, so hopefully now you've watched those videos and you know that an action potential involves voltage-gated sodium and potassium channels in the plasma membrane. Okay, now in neurons, these voltage-gated channels are on the axon and nerve terminals, but we'll worry about that later in the semester. In the case of muscle tissue, they are located outside of a structure called the motor end plate. Okay, and once again, remember that muscle tissue has exactly the same set of these voltage-gated channels. So the way things work in a neuron are basically the same way that things work in a muscle cell. Okay, in response to a big enough change in the resting potential of the membrane, they allow ions in and out. This is going to cause a much larger change in the membrane potential, as we'll see on the next slide here. Okay, so I wanted to put up a little bit of a mock-up, maybe a little bit simpler diagram, and see if this would also work for you. So there are some things that I want you to notice about this particular diagram right away. First of all, you've got voltage-gated sodium channels. Secondly, voltage-gated potassium channels. Remember, and I've put these on the channel so that you can see that sodium is a positively charged ion, and so is potassium. Now, as you can see in the diagram here, there's a lot more sodium on the outside than there is on the inside. There's also a lot more potassium on the inside than there is on the outside. Okay? Now, there is more sodium on the outside than there is potassium on the inside. And so basically, this results in a slightly lower charge on the inside of the membrane as opposed to the outside of the membrane. So basically, there's a charge of negative 70 millivolts on the inside. Now, I can't remember if I've used this example or not because I've had to re-record this slide several times, but I wanted to give you sort of an example or an analogy for this charge. So you may be wondering, how is it if we've got positively charged sodium on the outside and positively charged potassium on the inside, how is it that we have a negative charge? We're dealing with positively charged ions, right? So here's my analogy for this. Let's say that you have $100 and I have $30. We're both positive in terms of money, but compared to you, I have negative $70. I hope that makes sense, because there are positively charged ions on the inside of the membrane, but fewer of them, and this results in a negative 70 millivolt charge on the inside, but only as compared to the outside. Now, the fact that there's a negative charge on the inside and a positive charge on the outside means that effectively there are poles. You have a negative pole on the inside and a positive pole on the outside. And I want to take your memory back to those little bar magnets that you used in elementary school. So remember that they had a plus and a minus on them, right? For a positive pole and a negative pole. And if you look at any battery, you see a positive terminal and a negative terminal. And so those could also be called poles. And here you've got a negative and a positive charge, a negative charge on the inside, positive charge on the outside. And so these are called poles, and that means that this membrane can be said to be polarized. So right now there is a resting potential of negative 70 millivolts. That means that this membrane is polarized. Now let's take a look at what happens if you alter the charge on the inside. So let's say that some sort of disturbance has hit the membrane to where the inside charge has moved up to negative 55 millivolts. This is something that's called threshold. Okay, so at threshold, then a very important event is going to happen, and it's going to be that the sodium channel is actually going to open. So if you look at the sodium channel right now at negative 55 millivolts, the channel opens. Now, when this channel opens, there are a couple of things that are going to make these sodium ions come in really fast. Okay, one is their chemical gradient. There's more of them on the outside than on the inside. So even in the absence of electrical charges, sodium would come rushing in. But as we've mentioned before, there are also electrical charges in play. The sodium ions are positively charged. The inside is negative relative to the outside. And that means that not only diffusion, but electrical forces are bringing the sodium ions in. So let's bring them in. Remember that this is a polarized membrane, and now we're going to depolarize the membrane. We're going to get rid of this negative charge on the inside. So here they come. Now, when these positively charged ions move into the cell, they're going to make the charge rise. So the charge rises, and it actually continues to rise until it hits plus 30 millivolts. Now this process is called depolarization. You had a polarized membrane, now you've depolarized it. Okay, now at plus 30 millivolts, a couple of different things happen. First of all, the sodium channel will close. So if you take a look at that, it's closed. Now take a look at the potassium channel. At plus 30 millivolts, the potassium channel is going to open. So now it's open. And let's talk about what happens to the potassium ions. Now, first of all, they're going down a concentration gradient. So they're going to get out of there pretty quickly. They're going to move from the inside to the outside. But now that the inside of the membrane is plus 30, they want to move away from that charge. Positive repels positive, remember? So now when potassium moves out of the cell, it's moving down a concentration gradient, but it's also moving down an electrical gradient. So what's going to happen? They're going to move out. Now when these positive ions leave the cell, what's that going to do? If you lose positively charged ions, that's going to take the inside of the membrane back to negative. Okay, so because you're moving back to negative, and you will move back to negative 70 millivolts, and even beyond that, you're moving to repolarization. Okay, so when you move back to negative 70, you are repolarizing the membrane. Eventually, you're going to move all the way to negative 90 millivolts, and you haven't just repolarized. At that point, you have hyperpolarized. So during the process of hyperpolarization, or during the phase of hyperpolarization, what's going to happen? Well, the potassium channels are going to close. And then you're going to see a reset of the sodium channels. They're going to stay closed. But as you saw in the video, the leak channels and the sodium-potassium pump, those are going to swing into action again and completely reset your membrane. So that is an action potential. Okay, so here's your big picture. Resting state, the membrane is at negative 70. Now if you take a look at the number 1 down in the chart, well, first of all, let me explain what we're actually looking at here. So the y-axis is the membrane potential. So what you're seeing with this purple line, as it rises, that means that the charge is rising as well. Okay, the x-axis is time. So as you're moving from left to right, you're moving forward in time. Okay, so let's start at the resting state, which is number 1. You're at negative 70, you have a polarized membrane. Okay, so you move up to threshold due to some sort of disturbance. Okay, when you move up to threshold at negative 55, then at negative 55 all of the sodium channels will open. Sodium comes rushing in. When sodium rushes in, it's going to raise the charge on the inside of the membrane. This process again is called depolarization. So as the sodium channels are open, you go through phase number 2, depolarization. When you hit positive 30, those sodium channels will close, and so there's no more sodium moving at all. At this point, at this same point, potassium channels will open. Now when potassium channels open, then potassium will go down its electrochemical gradient, moving from inside to outside. So as those positive ions leave the cell, then you're going to see your charge move back down, and this is called repolarization. And so again, this is phase 3 of the action potential. Now once you get down below negative 70, and this will happen, you haven't just repolarized, you've hyperpolarized. So take a look at number 4. At number 4, it's at that point where the potassium channels actually close again. So now the sodium channels are closed, the potassium channels are closed, so there's no more ion movement through the voltage gated channels. And how do you get from 4 back to 1? The way that you get from 4 back to 1 is the leak channels and the sodium potassium pump that the video mentioned. So you get this complete reset to get back up to number 1 again. And so this whole process takes about 4.5 milliseconds. So it is something that happens really fast. Now I realize that the video that you saw showed the ions kind of slowly moving in and out of the membrane. That is the one problem that I've got with that video. The ions move really, really fast, and that makes this a really, really fast process. So the faster the diffusion, the faster the whole process of this action potential. And it happens very quickly in just about 4 to 4.5 milliseconds. Okay, so the next thing that I want you to do is to stop this video and play the neuromuscular junction excitation video. This is a summary of what you've just seen. So the events at the neuromuscular junction will produce an action potential on the skeletal muscle cell. Okay, so basically here's what happens. At the end of the axon terminal, I want to make it really clear that this is the neuron. The end of the neuron has voltage gated calcium channels. Now these open up, these cause calcium to come in, and what calcium will do to the neuron is allow it to carry out exocytosis of a neurotransmitter. So the neurotransmitter goes into the synaptic cleft and diffuses across the synaptic cleft. Once it has diffused, it'll bind to ligand-gated sodium channels on the motor end plate. Now, I want to make something really clear. This motor end plate is a portion of the skeletal muscle cell that does not contain voltage-gated channels. It only contains ligand-gated channels. Okay? So those ligand-gated channels will open in response to neurotransmitter binding. Okay? Now, once the neurotransmitter binds, sodium rushes in. Now, it will rush in, and so the area under the motor end plate, under that portion of the membrane, that membrane potential will rise. Okay? Eventually, that rise in membrane potential will reach the edges of the motor end plate. Now, at the edges of the motor end plate, you're actually going to see voltage-gated channels, and this is where those voltage-gated channels actually open up. Okay, so think of the motor end plate as this small sort of area where you've got ligand-gated channels. Now, this is an area that causes the disturbance that cause the voltage-gated channels around the outside of the motor end plate to get set off. Okay, so basically, when this membrane gets depolarized, what you're going to see is you're going to see that action potential sweep across the skeletal muscle cell. It'll go down into the skeletal muscle cell. Remember that the T-tubules are an extension of the cell membrane into the skeletal muscle cell. Okay, so when that action potential hits the T-tubules, it will also hit voltage-gated calcium channels in the sarcoplasmic reticulum, and in response to that charge, they'll release calcium from the sarcoplasmic reticulum. Okay. Now, in the meantime, back in the synaptic cleft, the neurotransmitter gets broken down. So without all of this happening, without the neuromuscular junction triggering an action potential on the rest of the skeletal muscle cell, then muscle contraction is not possible. Look at this. All right, first of all, let's zoom in on our neuromuscular junction. Okay, so as you can see, basically, this is where the end of the neuron actually meets the membrane of the skeletal muscle cell. Now, this is a little bit more of a zoom in on the neuromuscular junction. So we had a top-down view of it in the previous section, and here we're taking a look at the side view. Okay, so I want you to see what's going on. Now, the nerve impulse, which is also known as your action potential, when this comes down the axon terminal, look just to the left, you'll see voltage-gated calcium channels. Okay, now what these voltage-gated calcium channels are going to do is they're going to let calcium in when they get zapped with an action potential. Okay, at that point, these synaptic vesicles, which contain acetylcholine, are actually going to fuse with the cell membrane, with the very end of the axon, and release their neurotransmitters into the synaptic cleft. So let's take a closer look at that. Okay, so if you look at the very top, at step 1, acetylcholine gets released from the synaptic vesicle. Okay, in step 2, the acetylcholine binds to the acetylcholine receptor. Okay, now once that happens, that acetylcholine receptor will open up a ligand-gated sodium channel. Sodium comes into the skeletal muscle cell. Okay, step 3 happens when enough sodium enters at the motor end plate. It will bring the membrane up to -55 on the edges of the motor end plate. This causes an action potential everywhere else in the skeletal muscle cell. So you produce a skeletal muscle action potential. Okay? Now, you don't want acetylcholine to remain attached to the acetylcholine receptors indefinitely. Otherwise, sodium would continue to stay in there, and you would continue to have action potentials on the skeletal muscle cell. So basically what happens next is that acetylcholine gets broken down. And with acetylcholine getting broken down, this basically means that these 4 steps only produce a limited contraction, so something that's called a muscle twitch. For contraction to continue, this means that the neuron has to send another action potential, which will cause steps 1 through 3 to repeat. So when you contract a muscle, say when you contract your biceps brachii, you lift your arm, you lift your hand up toward, you flex your arm. This is the result of a whole lot of action potentials telling the muscle to keep contracting until you're finished. Okay, so this is just an actual picture of a neuromuscular junction, and had we been in lab, I would have been able to actually show you that on the slide. We've got a few really nice slides of an actual neuromuscular junction. Now at this point, what I'd like you to do is to play the remaining 2 Pearson videos, the excitation contraction coupling video and the cross-bridge cycle video, and we'll resume our discussion after you do that. Okay, now that you've done that, we're going to take a look at all of these different steps. Okay, so from the very beginning, from the neuron all the way to relaxation of muscle. So the first thing that happens is, of course, the nerve impulse arrives at the axon terminal of the motor neuron. So that's going to be the very end of the motor neuron. This causes the release of acetylcholine. Acetylcholine will diffuse across the synaptic cleft. It binds to receptors in the motor end plate, and this will cause a muscle action potential just outside of the motor end plate. Okay, now acetylcholinesterase in the synaptic cleft, it destroys acetylcholine, and so basically you're going to get one action potential out of this, but not a second one. To get a second muscle action potential, you're going to need a second neuron action potential. Now, once your action potential has gone down into the T tubules, which you can see as these folds down into the membrane, basically, if you look at the sarcoplasmic reticulum and you look at what those little triangles look like, they're now open. Those represent channels. So they're going to allow calcium to diffuse out into the rest of the cell. Now, there is something that I want to mention here. The concentration of calcium ions within the sarcoplasmic reticulum is about 10,000 times that on the outside of the sarcoplasmic reticulum. This means that diffusion is going to happen very fast. Calcium is going to flood the entirety of the cell very fast. And now you know why that's important. Because you want as much calcium getting out and hitting as many troponin molecules at the same time as possible. Okay? So the muscle action potential has caused voltage-gated calcium release channels in the sarcoplasmic reticulum to open. Next, calcium binds to troponin on thin filaments. And so what's going to happen is when calcium binds to troponin, as with every other protein, you bind something to a protein, it changes shape. You bind calcium to troponin, it changes shape. When troponin changes shape, it moves the tropomyosin that it's attached to. This exposes binding sites for myosin, and now actin is open for business. Actin is ready to be bound by myosin. Okay, at this point you go through the events of the cross-bridge cycle. Okay, you have myosin cross-bridging to actin. Phosphorus leaves, which strengthens your cross-bridge. ADP leaves. This shape change causes the power stroke, and then the thin filaments are pulled towards the center of the sarcomere. Now there's another picture here. On the sarcoplasmic reticulum, you've got calcium active transport pumps that are constantly working to try to bring calcium back into the sarcoplasmic reticulum. Okay? When the channels when the release channels are open, then they completely overwhelm the ability of the pumps to bring calcium back in. So calcium moves up very quickly. But if there's not an action potential causing the release channels to open, they're going to close right back up. So when they close up, the calcium active transport pumps can get going again, and that's going to pull calcium from the sarcoplasm back into the sarcoplasmic reticulum. So basically what you're doing is you're resetting your skeletal muscle cell at this point. Okay, now the result of bringing calcium back in is that the calcium in the sarcoplasm will lower. So when the calcium in the sarcoplasm lowers, then here's what happens. Calcium leaves troponin, so troponin will change back to its original shape, which means that tropomyosin shifts back over the actin, meaning that the binding sites for myosin are now blocked. Now when the myosin binding sites get blocked, sorry about that, when the myosin binding sites get blocked, then the muscle's going to relax, and here's the reason why. So if you take a look at the diagram of the sarcomere right above the number 5, you'll notice those little spring-like things that look like they're going from the thick filaments to the Z discs. So again, remember that the Z discs are those sort of zigzag looking things on the edges of the sarcomere. Now, if you look at the sarcomere diagram just above the number 6, then you'll see that those springs have been compressed as the sarcomere shortens. Now, in the absence of calcium, there's an absence of cross bridges. That means there's no contact between thick and thin filaments, and those springs are going to open back up. They're going to return the sarcomere to its original position, as you see above the number 5. So this is basically what happens. This is basically how you get muscle relaxation occurring.